Friends, welcome to our lecture on pharmacodynamics brought to you by Professor G N Purohit from GNP Sir classes. Watch the video completely to understand the key points in pharmacodynamics. So here are the contents on which we will discuss, from principles of drug action to the factors modifying drug action. So first we introduce you to pharmacodynamics. So first let's discuss the general concepts of pharmacodynamics. Pharmacodynamics is the study of physiological and biochemical effects of drugs and how these effects relate to a drug's mechanism of action. It focuses on the action and the effects of drugs within the body. In general, pharmacodynamics characterizes what a drug does to a patient. In contrast, the study of pharmacokinetics addresses what the patient's body does to a drug. Effective use of a drug requires knowledge of the drug's pharmacokinetic and pharmacodynamic properties. Now let's see what are the principles of drug action. Drugs, except those gene-based, do not impart new functions to any system, organ or cell. They only alter the pace of ongoing activity. The basic types of drug action can be broadly classed as depression, which means selective reduction in activity of specialized cells. Examples: barbiturates, quinidine. Drugs stimulating one cell type may depress another. The second type of drug action is replacement, which is the use of natural metabolites and hormones in deficiency states. Examples: insulin in diabetes and fluids in dehydration. Other types of drug action include stimulation, which means that the drug causes a selective enhancement of specialized cells' activity. Examples: adrenaline. Other types include irritation, which is non-selective effect on specialized cells, and cytotoxic effect, which causes a selective action on parasites or cancer cells. Now let's talk about the basic mechanism of drug actions. Mechanisms of drug action can be grouped as non-cellular mechanisms of drug action and cellular mechanisms of drug action. First, let us talk about the non-cellular mechanisms of drug action. Drug reactions that occur extracellularly that involve non-cellular constituents include the following: the physical effects. The examples for this include protective, adsorbent and lubricant properties of locally active agents. Then, other non-cellular mechanisms include the physiochemical mechanisms, for example surfactants, chemical reactions and modification of body fluid composition. The examples are magnesium sulfate, mannitol, and dextran. Now let's discuss the cellular mechanisms. These include 1. The alteration of specific cellular components, for example inhalant anesthetics. 2. Influence on membrane structure or function, for example local anesthetics. 3. Interference with neurohumoral transmission, for example recepine. And 4. Enzyme inhibition. Now we detail out the protein targets for drug binding. 4 main regulatory proteins are commonly involved as primary drug targets: receptors, enzymes, carrier molecules and ion channels. Receptors are sensing elements in the system of chemical communications that coordinate the function of different cells in the body, the chemical messengers being hormones, transmitter substances or other mediators. Receptors have a role in chemical communication. Receptors act as agonists or antagonists. The type of receptors include 1. Ligand-gated ion channels. 2. G-protein coupled receptors. 3. Kinase-linked and related receptors. And 4. Nuclear receptors. The receptors function to propagate, regulate or amplify the signals or adapt to changes to maintain homeostasis. Many drugs are targeted on enzymes. Most commonly, the drug molecule is a substrate analog that acts as a competitive inhibitor of the enzyme, either reversibly or irreversibly. The carrier molecules, often proteins, bind to icons or molecules for their transport across cell membranes. The ion channels are either ligand-gated or voltage-gated. Ion channel modulation by drugs, acting directly on the channel or indirectly, is one of the most important mechanisms by which pharmacological effects are produced at the cellular level. Now let us discuss some definitions and concepts in pharmacodynamics. The affinity is the tendency of a drug to combine with a particular type of receptor, whereas efficacy, also called intrinsic activity, is the maximal effect a drug can produce. The potency of a drug is the dose required to produce a particular effect. It is influenced by the drug affinity and is not correlated with efficacy or safety. Selectivity of a drug is the capacity of a drug to preferentially produce a particular effect. When all the effects produced by a drug are due to a single mechanism of action, the drug is said to be specific. An agonist is a drug that possesses affinity for a particular receptor and causes a change in the receptor that results in an observable effect. They are either full, partial or inverse agonists. A full agonist produces a maximal response by occupying all or a fraction of receptors. Affinity equals 1, efficacy equals 1, partial agonist produces less than a maximal response even when the drug occupies all of the receptors. A partial agonist has less intrinsic activity than a full agonist. Affinity equals 1, efficacy equals 0 to 1. An antagonist is a drug that blocks the response produced by an agonist. Antagonists interact with the receptor or other components of the effector. There can be competitive or non-competitive antagonism mechanism, but antagonists are devoid of intrinsic activity. Affinity equals 1, efficacy equals 0. Inverse agonist activates a receptor to produce an effect in the opposite direction to that of the well-recognized agonist. Affinity equals 1, efficacy equals -1 to 0. Drug action is the initial combination of the drug with its receptor resulting in a conformational change in the latter, in the case of agonist, or prevention of conformational change through the exclusion of the agonist, in the case of antagonists. Now let's discuss drug-receptor interactions. The drug-receptor interactions are clear by the figure, which shows that with agonists there is an activation response, whereas with antagonists there is a blockage response with finally no response. Binding and activation represent two distinct steps in the generation of the receptor-mediated response by an agonist. If a drug binds to the receptor without causing activation and thereby prevents the agonist from binding, it is termed a receptor antagonist. Receptor drug binding can be measured by radioactive traces, injection of isotope into animals, in vitro radio-labeled drug binding, and many other techniques. The forces that determine whether or not a drug will interact significantly with a receptor are thermodynamic and involve enthalpy, the absorption or production of heat associated with binding interaction. 5 types of chemical bonds can be formed between a receptor and a drug. They are covalent, electrostatic, hydrogen, van der Waals and hydrophobic. The relation between the response produced by different dosages is expressed by a graphical representation called dose-response curve, DRCs. These are of two types: SAD phase, I, graded dose-response curve, and E, quantile dose-response curve. It gives the relation between dose of a drug and the intensity of response in a single biological unit. The curve depicts that when the dose exceeds a critical level, threshold dose, the response also increases progressively until it reaches a steady level, ceiling effect, ceiling dose. The threshold dose may be defined as the minimum dose required to produce an observable response. The dose-producing ceiling effect may be called a ceiling dose, which may be defined as the minimum dose producing the maximum response. When the graded response is plotted as a graph, a hyperbolic curve as shown is obtained. The quantile dose response represents the percent response of animals in a group of population to the doses of a drug. LD50. It is called the median lethal dose, which is defined as the dose that would be expected to kill 50% of the exposed population. ED50. It is called the median effective dose, which is defined as the dose that would be expected to produce a desired therapeutic response among 50% of the exposed population. Both the two estimates are used for the assessment of the safety of a drug by determining the therapeutic index, which is the ratio of LD50, ED50. Wider the ratio, safer will be the drug. When two or more drugs are given simultaneously or in quick succession, they may be either indifferent to each other or exhibit synergism or antagonism. When the action of one drug is facilitated or increased by the other, they are said to be synergistic. Synergism can be additive or supra-additive, potentiation. Examples of additive effect is aspirin plus paracetamol equals as analgesic antipyretic, and an example of potentiation is acetylcholine, H, plus phosostigmine equals inhibition of H breakdown. When one drug decreases or inhibits the action of another, they are said to be antagonistic. The antagonism can be physical, chemical, physiological, and receptor block antagonism. A variety of factors either singly or in combination influence the magnitude of response observed following the administration of a drug. We will discuss them. Species variation. The response of a drug is not uniform among different species. For example, morphine produces CNS depression in men and dogs but excitation in cats. Rabbits can thrive on belladonna leaves which are too toxic to other species. The effect of a drug may vary with the age and sex of the animal and also the body weight. The time of drug administration may influence the response. The route of administration also determines the speed and intensity of drug response. As shown, the intravenous route has the fastest response, followed by intramuscular, intraperitoneal, subcutaneous and oral routes. The efficacy of a drug can be affected by patients' beliefs, attitudes and expectations. Placebo, a Latin word meaning, I shall please. Placebo is an inert substance that is given in the replacement of a medicine. It works psychologically rather than pharmacologically. It is important to remember the pharmacodynamic and pharmacokinetic interactions. Pharmacokinetic interactions refer to interactions between the drugs during absorption, distribution, metabolism and excretion processes, resulting in either increase or decrease in the concentration of the drug at the site of action. Some of the examples are shown that may alter the drug absorption. Weak and debilitated animals are more sensitive to drug effects than normal healthy animals. Tolerance is a widely occurring adaptive biological phenomenon. Drug tolerance may be natural or acquired. In natural drug tolerance, an individual species is inherently less sensitive to the drug. For examples, rabbits are tolerant to atropine due to the presence of atropinase enzyme. Chicks are tolerant to strychnine due to the underdevelopment of the spinal cord in chickens. Acquired drug tolerance occurs by repeated use of a drug in an individual who was initially responsive. Lastly, we discussed two terms. When certain drugs are administered at the same dose at frequent short intervals, the response will decrease progressively. This is called as tachyphylaxis. Drug resistance refers to the tolerance of microorganisms to the inhibitory action of antimicrobials, e.g. Staphylococcus to penicillin etc. Thank you very much for watching this online lecture. Don't forget to like, share and subscribe to GNP Sir Classes official channel. Bye bye.